Hello and welcome to the Plant Breeding and Genomics Community of Practice webinar. My name is David Francis. I'm the co-leader of PBG and I'll be serving as your host today. This is a special presentation on soil-borne diseases of dry bean presented by Dr. Martin Chilvers of Michigan State University's Department of Plant, Soil, and Microbial Sciences. He earned his bachelor's degree with honors and a PhD from the University of Tasmania, Australia, with subsequent postdoctoral training at Washington State University. Dr. Chilvers leads a field crop pathology program encompassing both basic and applied research, with current emphasis on seedling and root rot diseases, Pythium, Phytophthora, and Fusarium species. Dr. Chilvers is a plant pathologist who works closely with plant breeding teams from Michigan to East Africa. He will be speaking about identifying soil-borne diseases with morphological and molecular techniques. After the presentation, you will have a chance to ask questions. If you have a question, you can simply type it into the question box on your screen and hit return. If you can't see the question box, click the small plus sign next to the word question on your control panel to open it up. We will answer as many questions as we have time for in the question period. Now I'm going to switch control of the screen over to you, Martin.
Thanks, David. All right. Okay. Well, good morning or good afternoon, depending on where you are. As David said, my name is Martin Chilvers, field crops pathologist at Michigan State University. I also am quite fortunate is that I have a colleague who also works on dry beans and sugar beets, and that's Dr. Linda Hansen here with the USDA ARS unit based at Michigan State University. So I just want to acknowledge her as well. Okay, so today what I wanted to do, this is really the first plant pathology themed webinar for PBG. So I want to give sort of an overview of soil-borne diseases, and we may need to follow up with some more specific webinars in the future. What we're going to do today is talk about some of the major players and the symptoms that they produce on dry beans, some isolation methods and techniques to get at those particular species, how we go about morphologically identifying those in an overview, and then some molecular identification techniques and tools that we currently use. In talking about root rot diseases, they certainly present something of a challenge, especially when compared to foliar diseases. Foliar diseases are typically pretty easy to identify and isolate a causal organism. It's right up there in your face on the plant, very easy to walk through a crop, get an idea of the incidence and severity of disease. When we're talking about root rot diseases, of course, everything happens below ground, so we're pretty much sort of talking about a black box, and so it can be quite difficult to sometimes establish a causal agent. It can be difficult to collect incidence and severity data, and of course all of this can be quite labor intensive too. So what we're doing in my lab is using a variety of different tools to try and open this black box, to have a look at what's happening inside at the root zone, both at the seedling and later stage root rot disease. So we use a number of different tools, of course, plant symptomology, including above ground and below ground symptoms, isolating the causal organisms, collecting populations, performing Koch's postulates to see which species is actually causing disease, pathogenicity screening, both from the sense of which particular species is causing disease, and then also working with breeders to try and identify resistance to the particular pathogenic organism. And in our identification efforts, we use a lot of DNA sequencing, and we also use real-time PCR for tracking root diseases over time in the root system. And we're also getting into microbial community analysis, which I'll touch on at the end of this presentation. So when we talk about dry bean root rot pathogens, there are quite a few, but there are sort of three major recognized groups, and they are Rhizoctonia solani, Fusarium species, and Pythium species. Here's an example of the amount of damage that can be done by Rhizoctonia solani. This is from last year, so 2013 field trial that we set up, trying to have a look at yield loss to both Rhizoctonia solani and Fusarium virguliforme. As you can see, the left image there is a control plot where we've used the dry bean Zorro, and the stand looks pretty good, everything's pretty much okay. And then on the right, you can see where we've inoculated with only 4 grams of inoculum per foot of Rhizoctonia, and we've pretty much killed everything. We've got maybe two or three surviving dry bean plants. When we dig these plants up and then trying to identify Rhizoctonia in the field, what we're looking for are small sunken lesions which progress to cankers, typically with reddish-brown borders. And I'll touch on the different subgroups of Rhizoctonia solani, but there are a number of different anastomosis groups, and some of these have different host ranges and also different stages that they infect. Some prefer seedling, some prefer later season or cause later season root rots. So we can see a lot of seedling damping off from these particular Rhizoctonia solani, as well as later season root rots. And seedlings are most susceptible to Rhizoctonia. Fusarium root rot is a very large group, or describes a very large group of potential Fusarium that may do damage to dry beans. One of the most recognized is Fusarium solani forma specialis phaseolae, or Fusarium phaseolae, depending on which naming scheme you're currently using. There are also several other species that definitely do cause a lot of damage, and one of those that we're currently looking at is Fusarium virguliforme, very closely related to Fusarium phaseolae. And with Fusarium root rot, we get reduction in root mass, we get sometimes stunting and yellowing. Roots will have reddish lesions with a sort of dry rot type appearance. And the other type of sort of Fusarium disease is what we might call Fusarium yellows or Fusarium wilt. And this is typically caused by Fusarium oxysporum forma specialis phaseolae. Here's an example of some greenhouse work where we have inoculated this Zorro dry bean with some Fusarium virguliforme, and you can see that the taproot has been severely affected. These plants are compensating by sending out adventitious side roots to compensate for that loss in taproot. And then the third major group are the Pythium species. Again, just like the Fusaria, there are a lot of different species here. They can cause damping off or root rot. They are quite different though, of course, because these are oomycetes and not true fungi. So they really do prefer high soil moisture. They form sporangia, which release zoospores, and those zoospores have chemotactic abilities, so taxic or other abilities. So they're able to swim towards the plant roots in response to any exudates that the plants are giving off. And they produce typically water-soaked soft lesions. And there are a number of other fungi as well that can cause seedling diseases or root rots, later season root rots, including Aphanomyces eutyches, another oomycete, which requires high moisture, Thalaviopsis, which is a warm and wet pathogen, perhaps more so in the subtropics to tropics. And there are other species such as mucor that are not particularly well understood. A lot of these type of species we might kind of consider to be secondary, but there's increasing evidence that they play a, excuse me, a large role in root rot. Excuse me. And then there's a sterile white Basidium mycete as well, which is also gaining some recognition as being a root rot pathogen. So we've described 3 major classes, but very often a root system can be attacked by multiple pathogens at the same time. So we end up having a disease complex. In addition to this, many of these species are potentially synergistic, so that damage from either alone is greater when both are together. And of course, when we're dealing with complexes and synergism, it can be very difficult to diagnose, and symptoms can really vary with different climatic conditions and different combinations of pathogens. Here's another example that might not be completely uncommon either. This is a situation that I was called out to last year. This particular commercial grower had planted or resown this dry bean field, and even at the second planting he completely lost that field. So it took a little bit of sleuthing around to really figure out what was going on. Disease at this level across the entire field is quite unusual for a root rot pathogen, particularly given the weather conditions that we had had. And what we ended up figuring out that this particular issue is caused by herbicide damage, some herbicide that had been residual on the soil surface. So that's something else we need to really keep in mind, the abiotic factors that may be causing what appear to be seedling diseases. And we shouldn't forget as well that there's always the potential for herbicide and pathogen interactions, and this is quite well documented in a number of different systems, where pre-emergent herbicides may do some damage to the seedling and predispose it to infection by pathogens. So this is something else to keep in mind when we're trying to identify a causal organism. And this is just an example of that. The same cultivar, same planting date, just in a different field, a neighbouring field that was planted by that same grower. So really, to get to that conclusion that it was a herbicide that was doing the damage, we had to really work with the grower, have a look at his spray record, see what the differences were between fields. Unfortunately, he had planted this same field the very same day, so we were able to go through and figure out that it really was that herbicide causing the damage in that previous field. Okay, so let's talk then about what we would want to do if we come across a field and we want to do some isolation to determine the causal organism. Okay, so first we want to try and remove as much soil from the roots as possible. Typically in our lab, we take the roots, place them in a mesh bag, such as an onion bag, something similar, and then we place them under running water for about an hour to really remove all of that soil, and sometimes working it with our hands to really remove a lot of that soil. Really essential to get all of that soil away, because of course that soil contains a lot of microorganisms and they're going to grow on your plates and contaminate the culture process. If you're dealing with older tissue, it might be possible, excuse me, to scrape back the epidermal layer and then by that process remove contaminants that are not really causing the disease. And of course, we want to isolate from the advancing lesion front. So we want to have tissue that is beginning to show disease symptoms, not tissue that is completely dead already. Obviously, isolating from tissue that is completely dead, we may very well be isolating saprophytes or secondary invaders that have also colonized that tissue. So it's very important to have a good sample collection to determine what's really causing disease in the field. And then also we use a lot of surface disinfestation. Typically this just consists of a weak bleach solution, something like a 0.5%. This is quite handy for isolating fungi. However, you should really use caution if you're trying to isolate oomycetes such as Pythium or Phytophthora, because they can be quite sensitive to the bleach. So in that particular situation, we don't typically use a bleach. We just wash really, really well and then use sterile paper towel to wipe off the moisture from those root systems, and then place small pieces onto agar. Media selection can have a huge influence on the composition that you recover from diseased dry bean roots. Depending on what you want to do or what you believe to be the causal agent, it will certainly influence the media that you select for isolation. If you're after a total composition, water agar can be quite useful. It's generally not too selective. However, you may very well get selection of fast-growing organisms. So, for example, if we were after true fungi and we weren't particularly interested in Pythium, we might want to think about adding some metal axle to that water agar to really reduce the amount of oomycete competition. In the lab here, we've done a number of projects where we use an oomycete semi-selective medium, which could be a base of something like cornmeal. We also use V8 agar for Phytophthora. And then we supplement that with a number of antibiotics. So the antibiotics are to remove secondary organisms like bacteria. So we use some pumarasin, ampicillin, and rifampicin to try and remove those. And because we're targeting oomycetes, we're not particularly interested in the fungi that are present. So we use some PCNB and some Benamil to help reduce the amount of fungal contamination, so that we can really target the Phytophthora, the oomycetes. For Phytophthora, Phytophoras tend to be slower growing than Pythium species, so if we're after that particular genus, then we'll want to think about including something to limit the Pythium, which are quite fast growing. So hymexazole is an agent that will really limit the amount of Pythium growth and help in the isolation of Phytophthora if that's something that is of interest. And for Rhizoctonia, this past season we used some Co and Hora medium, a little bit expensive but quite good for isolating Rhizoctonia. In our hands we found that quite useful as a semi-selective medium for the isolation of Rhizoctonia. Here's a typical setup in a laminar flow cabinet. Of course, anyone with any pathology background will be quite familiar with this. So, of course, to minimize secondary contamination, we work under sterile airflow. In this example, we've got paper towels, sterile paper towels ready to go to blot plant tissue. We'll typically have beakers sitting ready with washes of sterile distilled water, as well as a bleach treatment if that's part of the protocol. So now that you've got some isolates to work with, you're starting to get some growth from root tissue perhaps, we want to think about how pure that culture is. So does the culture look pure to you? Do you have multiple species coming off that particular piece of root tissue? And so some things that we do to try and purify cultures, because of course we want to get to the point of being able to identify individual organisms, we do things such as hyphal tipping or single sporing. So with hyphal tipping, you want to be very careful and make sure that you really are truly taking the hyphal tip from the growing colony of interest and not lumping together 2 or 3 different organisms. We want to be working with pure cultures for identification. If you have a fungal structure that's producing spores, or fungal colony rather, that's producing spores, we could potentially use single sporing methods to get at a pure culture. There's different methods that you can use. We sometimes do something analogous to a 16 streak that some of you may remember from microbiology classes, where we streak out bacteria of interest, sterilize our implement, streak it again, sterilize and streak again, and by that way we have essentially a gradient of the organism of interest. And so we can do this to go back and then after an overnight incubation look for spores that have begun germinating, and look for areas on the plate too where we have single spores that we can go in and only collect a single spore rather than a mass of spores, because the whole point is to get at a single pure culture. Typically we'll use some very stiff medium for this, so we'll increase the agar content perhaps to a 3% water agar mix, for example. That allows us to really go in and pick up the spore on the agar surface. Softer media just makes it a lot harder to sometimes pick that up. And you could potentially also do a spore suspension and then plate that out and look for do a dilution series so that you've got single colonies growing that you can collect pure cultures from. Okay, so now that we've got our pure isolates, we might want to store those, particularly if we're doing a large population collection. And there are a number of different techniques that can be used depending on the organism that you're working with. A couple of things that we've used in the lab are slants for both fungi and oomycetes. We typically store our slants in our hemp seed vials. So this is just a little vial with some water and hemp seed in it, and an agar plug of the culture of interest. This is our slant with the culture of interest just placed inside that slant. For oomycetes, we typically store ours at slants and hemp seed at room temperature. Oomycetes really don't seem to like the refrigeration. However, if we're working with true fungi, refrigeration might be perfectly fine. So you need to kind of think about the organism you're working with and potential method. Also for fungi, another method that's quite space efficient is a filter paper method. A number of labs use this. So basically you grow your fungal colony across a piece of filter paper that you've placed on the agar medium, let it become colonized, hopefully get lots of spore production on that piece of filter paper. Then you can lift up that piece of filter paper, and we use coin envelopes to store those in. Very important to dry those down, so place them over some dry rite or other sort of desiccant to remove the moisture, and then they can even be stored in the freezer at -20. And that works for quite a number of years. Okay, so now we've got our organisms of interest. How are we going to go through and do some morphological ID? And here I'm really just going to give you an overview. This is really no substitute by any means for a mycology class, and if you're planning on doing this sort of work, I'd highly recommend you take a mycology class. So the general sort of procedure then in identifying, say from a population collection or maybe even a diagnostic sample where we've collected 10 plants and we've got now 20 different plates we want to look at, we really want to begin by organizing the Petri plates of pure cultures into groups based on gross colony morphology. This will help us get an idea of the number of organisms that we're potentially dealing with and the incidence. We need to keep in mind that the media type that the isolates, the oomycete or the fungi are being grown on will really influence the colony morphology that develops, as well as the age of the colony. Fresh colonies might not have developed fruiting structures or sclerotia or even pigmentation that older cultures might develop. So we need to really be working with cultures of the same age. Our first step then, once kind of grouping our plates into different groups, is to go through and examine hyphae, fruiting structures, and for the potential of sclerotia production under the dissecting scope to really have a look at what we have there at that macro sort of level. And then once we've sort of confirmed our different groupings perhaps, then we can begin preparing some slides to really start looking at any fruiting structures or hyphae under the compound microscope. So I'll just show you some examples then of those three major groups. So this is Rhizoctonia solani. We've got a tan colony here, and we've got the initiation of sclerotia just beginning to develop throughout the plate here, these little dark structures. Rhizoctonia doesn't produce any asexual spores, so when we look at that underneath the compound microscope, we won't find any spores, of course. However, it does have very distinct right angle branching of the hyphae. So you can see this hyphae here and a very distinct right angle T branch. What's also very distinct is the constriction at the T junction and the septae just after that T junction too. So this is a nice microscopic characteristic that helps identify Rhizoctonia solani. Now, Rhizoctonia solani, as I mentioned before, are also divided up into anastomosis groups. And a number of people are working on these anastomosis groups and doing further sort of phylogenetic and taxonomic characterizations of these. And what we mean by these anastomosis groups is basically whether two isolates, when they're put together, can pair and confuse. If they're able to fuse together without any cell death, then we've got isolates that belong to the same anastomosis group. This is an example that Linda provided me demonstrating two different anastomosis groups. So we've got one anastomosis group along the top here and then a second one along the bottom. And what you can see is when these isolates have sent out hyphae towards the other culture, or towards the other isolate rather, where they've fused, we've got cell death occurring. So you can see within these hyphae we've got loss of cytoplasm, and those hyphae are dead or dying. So in this particular example then, we've got two distinct colonies, and they're obviously of two incompatible anastomosis groups. So that's how we go about testing for anastomosis groups and further subdivision within the Rhizoctonia solani group. This is an example of Fusarium. This particular one is Fusarium virguliforme, something that we're working with quite a lot in the lab. As I said, it's very closely related to Fusarium phaseolae. Typically with Fusaria, we get wet sporodokia production, and if you were to take a scalpel or a needle, you can go through and pick up some of that sporodokia and place it onto a microscope slide. Fusarium are also typically quite colourful in culture, so in that sense they're quite a lot of fun to work with, and that can also help in identifying the Fusarium species that you're working with. So here we've taken some of that sporodokia and plated, streaked that onto a microscope slide. And so this is demonstrating the very characteristic Fusarium macroconidia that are banana shaped. The spore size, length, number of septae differ between species. Sometimes to encourage fruiting and for differentiation of species, we may need to use some very defined medium so that we get reproducible spore size, spore production. And one of the very common media that is used is carnation leaf agar, and that's used in a lot of descriptions of Fusarium. Here's a Pythium culture. So what this particular Pythium species is very flat in culture. Some can be quite floccose or fluffy. Just kind of depends a lot on the species. And also in this identification process, you know, we can tell a lot by the media preference that we have for the different organisms. For example, one of the organisms we work with a lot in the lab is Phytophthora sojae. This isn't a dry bean pathogen, but it's a pathogen of soybeans, a very host-specific pathogen of soybeans. This particular species just does not grow on potato dextrose agar. So we can even use that as another distinguishing characteristic if we're having trouble distinguishing Phytophthora sojae perhaps from a Pythium culture. With the Pythiums, very often we'll get sexual oospore production. Many species are homophthalic, so we don't need a second pairing isolate to get that sexual reproduction structure. And then we'll very often get sporangia produced too. These are very lobate sporangia. We may need to use particular conditions depending on the Pythium species, such as grass blade culture, to really get the fruiting structures so we can go through and identify the different species that we might be working with. So let's talk then about how we might use molecular ID to supplement or extend the identification that we might do with morphological methods. So why would we want to use molecular techniques? Well, it's not reliant on morphology. And this is very useful if we've got traits that are limited or plastic. So if we've got a limited trait, what I'm talking about there is, you know, perhaps having difficulty in even production of spores. It's going to be very difficult for you to differentiate various species if you don't have spores to work with. The other example is perhaps we have very plastic spore size and shape. So it can be very difficult to differentiate, again, between two closely related species. Obviously, this has also helped us a lot with, you know, instances of convergent evolution where we've got similar sorts of structures, but the origin of those species is quite distant and they've diverged quite a long time ago. And it can be very useful for resolving relationships between very closely related species. And it may help support or resolve morphological taxonomy. And here's an example within the Fusarium species. This is relatively new, so quite a few of these may still be referred to as Fusarium solani or Fusarium, sorry. Okay, oops, sorry. Whoa. Okay, so in this example then, what I'm trying to demonstrate is the overlapping morphology between various Fusarium species. So this is from an example of Aoki et al. 2003. So what we have here are 3 different Fusarium species, and what we can see is the overlapping length and width of those macroconidia. So when we're having a look at this Fusarium chukemani, the size overlaps with the other group, this Fusarium virguliforme group, which also overlaps with the Fusarium phaseolae group. So that just kind of demonstrates to you some of those plastic characteristics I was talking about, or very similar morphologies between different species that molecular techniques really help to resolve. So if we're going to do some molecular identification, how are we going to do that? Average fungal genomes are about 35 megabases in size. We need to ask, are primers available to amplify the organism of interest? If we're working with an oomycete, what does the literature say about that? Or if we're working with a Rhizoctonia, what does the literature say about that? We want to think about when we're selecting loci for sequencing, can they be sequenced easily? Are there primers available? Reference sequences, of course, are incredibly helpful. If we don't have a reference, it can be quite hard to compare our work to others. So what database entries exist for this particular group that we're working with? And finally, morphological identity should really direct sequencing efforts, and we should never really lose that connection between morphology and any molecular efforts that we move towards. So a very, very common loci that is used, both in fungi and in oomycetes, is the internal transcribed spacer region. So this falls within the nuclear ribosomal DNA, and it consists of tandem repeats. So typically within a fungal genome, we're probably looking at perhaps 70 to maybe 200 copies, tandem copies of this particular locus. Because of that high copy number, it's quite easy to amplify. You don't need a great deal of DNA to amplify it, which can be helpful at times. But more importantly, we have some very conserved regions that we can design primers to. So we have the 18S and the 28S ribosomal regions. And because these are quite conserved, we can design primers to amplify this internal transcribed spacer region, which, because it doesn't code for anything, it evolves relatively quickly. And some very common primers that are used are ITS6 or ITS1 and ITS4 to sequence across this particular locus. And it's also very useful because it's about 500 to 600 base pairs in size for fungal organisms, and about 800 to 1,000 depending on the particular species of oomycete. So it's quite a manageable size to amplify and sequence. So because of these characteristics, ITS has become the default barcode. We have plenty of conserved primers that are available to work with across different taxa. As I mentioned, it's very easy to amplify because of its multi-copy nature and those conserved primers, and it's an ideal size for amplifying and sequencing. However, we do need to point out that there are some potential drawbacks here. Sometimes especially within fungi, there is very little variability or to no variability within ITS between very closely related species. So it might get us down to the genus level and perhaps some possible species, but differentiating between very closely related species within fungi, ITS might not give us that resolution to do that. And in that case, we need to look for additional loci. And again, that needs to, we should base that selection on previous work that's been done. And there are a number of examples of very commonly used protein coding loci that are used to do further identification. When we've got our sequence data back from the sequencing facility, we then want to go through and analyze that. And of course, it's only useful if you can compare it to something else. And we use GenBank for this, hosted by the National Center for Biotechnology and Information, which is an incredible resource. However, there is no third-party curation. So this means that there is a lot of error in the sequences or sequence annotation that has been submitted. So anyone listening today could go out and sequence something, identify it, and then submit it to GenBank without any additional curation, which is helpful, but also very problematic. And within GenBank, I believe the number is about 40% of GenBank entries for fungi are incorrectly labeled. So we need to use a lot of caution when we're using this resource for identification of species. Now there are efforts in different communities to develop improved databases, and this is just getting better by the day. If you're working with Phytophthora, I'm quite fortunate because there's a Phytophthora ID database, and I'm sure we'll have a Pythium sequences uploaded to this very soon, and that can be very helpful. So this database is curated. They typically require voucher specimens to be deposited, and so that's very useful as well, because if we have an issue with a particular isolate that's sequenced, we can go back to that vouchered specimen and really have a look at, is this the same organism or is it a different species? Do we potentially need to name a new species? And within Fusarium too, there's a Fusarium ID database. And within the Fusarium ID database, they've got a number of different loci. Within the Fusarium, ITS doesn't really give you much resolution within species, so they have a number of other loci, such as elongation factor and beta tubulin genes that you can go and sequence and then compare to their database. We are pretty fortunate with the oomycetes. ITS does typically give species resolution, and I'll mention that again in a slide that's coming up. And then when we're using these databases, we're typically using the BLAST algorithm, which just stands for Basic Local Alignment Search Tool. So it's basically a way of searching very rapidly against a large database of subject sequences, and it's a tool that has been used enormously throughout the bioinformatics community. It has been very, very instrumental in the comparison of sequences. And for more information on this, because we don't have time today, certainly log into GenBank, have a look. There's plenty of BLAST instructional information and videos that you can look at to understand how BLAST works and how you can use it for identification. Okay, so I wanted to just spend a couple of minutes just describing how we've used some of these techniques to identify Pythium or oomycete species that cause soybean seedling disease. I know this is a dry bean webinar, but this panel of oomycetes that we've recovered, I believe, is quite representative of what's out there across the Midwest region, and we're also using this panel to have a look at pathogenicity of various Pythium species to dry bean. So what we did is a survey of soybean seedling diseases. So we went out, our collaborators across the 12 states pictured here, went out and collected diseased soybean seedlings, then took them back to their lab, went through the washing process to remove all of the soil from that diseased seedling, and then plated a piece of the diseased tissue onto that semi-selective oomycete medium. From this, they sent us cultures from each of those 12 states. We took those cultures and placed them into hemp seed vials or cornmeal agar slants. We then increase the oomycete biomass for DNA extraction, and we typically in the lab here are doing still culture for that, so still liquid culture, and that seems to work very well for our oomycete as well as the Fusarium that we're currently working with. From there, because we had over 3,000 isolates, we used a robot to do the DNA extraction for us. We lyophilized the mycelia of those different oomycetes, and then used this autogen phenylchloroform robot to do the DNA extraction for us. And then, of course, once getting DNA back, we check that for quality prior to proceeding. Then we've used ITS primers 6 and 4 to amplify the ITS locus, and then submitted that for Sanger sequencing. And so we're getting sequences, between 700 to 1,000 base pairs in size. And I apologize to those that can hear the train passing by my office. Okay, so now that we collected all these ITS sequence data from these various oomycetes from across the Midwest, we wanted to identify what we had, okay? And to do that, we didn't use GenBank because of those inherent problems that I was talking about. We were quite fortunate because very recently, this particular paper, Robidal et al., had done a lot of work looking at sequencing ITS and the COX loci from the mitochondrial genome for identification of oomycetes. So they had a very nice indexed set of sequences that matched vouchered oomycete specimens in culture collections. And they did a lot of morphological as well as the molecular work to pair those things up and have a look at the relationships. So we were able to use this curated data set to identify our particular sequences that we collected from the different species. And this is just to show you a result from that particular soybean survey. So on the left there, we've got our frequency distribution from our collection in 2011. So we had 52 different Pythium species, 3 Phytophthora, 2 Phytopythium, and 1 Aphanomyces species. And in 2012, that's shown on the right there, you can see some variation from year to year. Pythium sylvaticum seemed to be quite prevalent in both years. However, the Pythium upapillum, which was second most abundant in 2011, was quite reduced in 2012, and we believe that was just due to the difference in environmental conditions between years. So anyway, this is really just to demonstrate how powerful these tools can be and the data sets that we can potentially generate. This identification, this molecular identification, has really allowed us to figure out which species are occurring where and at what frequency, and we've paired this with a lot of pathogenicity screening, so we have some understanding of how pathogenic these various species are. So information about incidence and pathogenicity to really try and determine which species are really causing root rots and seedling diseases out in the field. Okay, one other area that I wanted to talk about is the use of real-time PCR for quantification and for working on root rot pathogens or seedling diseases. And so we've been using this, we've been developing assays for soybean sudden death syndrome, which is a pretty devastating disease of soybeans. It's spread across most of the soybean producing states in the US, and it's also a pretty major problem in South America too. This is just a distribution of Fusarium virguliforme causing SDS across Michigan and where we found it. And for those of you that are not familiar with dry bean production in Michigan, we produce a lot of our dry beans in the thumb area of Michigan. So a lot of our acres that we're producing our dry beans in are also, we've also found this soybean sudden death syndrome. Here's just a couple of slides of that soybean sudden death syndrome in soybeans. So in this particular field, this grower lost 50% of his yield, and here's some of those more sort of typical symptoms on soybeans. I'll skip over that slide. So it's a two-stage disease. So it's very closely related to Fusarium phaseoli. So it has quite a lot of similarity with that dry bean pathogen, that dry bean root rot pathogen. It causes rotting in the taproot. It can reduce root mass. So this is a disease compared to a healthy taproot. And then the second stage of the disease is the fact that the fungus produces a toxin, and that's a dramatic foliar symptoms that you were seeing. So the fungus doesn't move above the soil line, but it produces a toxin that kills those leaves. And here again, it's just that demonstration of where we've confirmed SDS and where our dry bean acres are in Michigan. So we asked the question, what's happening with Fusarium virguliforme on dry beans? So we planted some dry beans in amongst our soybean trials at this highly infested location. We then use real-time PCR, which is just basically a technique where you essentially can monitor the amount of PCR product in real time, and using standard curves you can actually determine the starting amount of DNA in your samples based on the probe of interest. So in this mix of soybean and Fusarium and whatever other microbes are present, we can determine in that root sample how much Fusarium virguliforme is present. So on our y-axis here, we've just got the quantity of Fusarium virguliforme DNA represented in femtograms per gram of root tissue. And these are different market classes of commonly grown dry beans. What we noticed is that Red Hawk was extremely susceptible and had a lot of Fusarium virguliforme biomass or DNA in its root system compared to the other species. And so we took this a step further and also had a look at the nematodes that were present. There's some speculation that with soybean sudden death syndrome that the soybean cyst nematode can potentially exacerbate disease. So we just had a look at that in dry beans. And although there wasn't a tight correlation, you can see something of a trend. That Red Hawk variety that had a lot of Fusarium virguliforme also had a lot of lesion nematodes, which are demonstrated in the red bars here. Whether this is just susceptibility of the Red Hawk variety to lesion nematodes as well as the Fusarium virguliforme, or if there's a correlation there, we're not too sure yet. Or sorry, a causal relationship between the two, we're not quite sure, but that's something that we're currently working on. And this is just a photo of that soybean sudden death syndrome, initial symptoms on soybean and on a dry bean plant grown in the greenhouse. We haven't to date seen this very typical symptom out in the field on dry beans, but we can get it in the greenhouse. So we are quite concerned about this pathogen and what might be to come yet for dry bean producers. And last year we did an initial dry bean yield loss study where we had controlled inoculated or not inoculated plots. And overall we were seeing some yield loss where we had inoculated those dry bean varieties with Fusarium virguliforme. Okay, I just wanted to finish by mentioning next generation sequencing. I'm sure if you're involved in breeding at all, you'll certainly know about this technology. So what I've been talking about up to date is the use of Sanger sequencing, which is essentially, you generate PCR product and then get returned a single sequence of typically about 800 base pairs in length. So it's very low throughput in comparison to next generation sequencing. Next generation sequencing allows us to generate millions of reads and at the base pair cost at a fraction of the cost of Sanger sequencing. So this is, of course, has led to its use in a number of different areas, and it really is changing how we do microbial community studies. And of course, if you're in the breeding area, genotype-based sequencing is also something of interest. So instead of using microsatellite repeats or something like that, we can just sequence the entire genome of our plant of interest, perhaps to identify SNPs that vary between cultivars. So in terms of root rights, well, we're currently looking at using this technology to do metagenomics or microbial community analysis to really get a better understanding of what's present in the root tissues. Bacterial tools for handling data are pretty well developed, and there are a number of efforts to develop tools and techniques to look at the fungal and oomycete populations that are present in roots and soil samples or other environmental samples. And I'll just end here, and we're pretty much out of time, with some additional resources. For field identification, there are some really nice resources. Something that we're currently developing as part of a NIFA project with e-extension is beanipm.pbgworks.org. And this is a website that's mobile friendly, hopefully for use in East Africa, so that can be accessed on on smartphones and used for identification of pests and diseases in dry beans. So if you have any feedback on that, please let us know. We'd like to hear from you. And that was generated from some information that was developed through the IPM pipe. So legume IPM pipe is another source of resources. And the University of Nebraska-Lincoln has a really nice document looking at these various root rot diseases out in the field and how to identify them in the field. And with that, I'd just like to acknowledge my lab and the various funding sources, and I'd be quite happy to take some questions.
Thank you, Martin. We've got about 10 minutes to answer questions. For those of you who missed the beginning of the presentation, you can use the question box on your screen to type in questions and hit return. If the question box is closed, you can click on the small plus sign next to the word. That will open up the question box. I'll be reading the questions out loud, and we'll answer as many questions as we have time for. So Martin, there was a question that came in during the course of your talk, and it had to do with the anastomosis groups.
Yeah.
It might be helpful to go back to that. So this was a clarification question, and the suggestion was that unrelated Rhizoctonia won't fuse at all. If they are essentially the same strain, when they do fuse, the hyphae won't die. If they're the same AG but not the same strain, then cell death may occur. Is that
sure?
Is that an accurate?
Yeah, absolutely. That's correct. So that's Linda Hansen, my USDA colleague, who has a lot of experience with Rhizoctonia. And I apologize for misspeaking on that particular area.
Okay.
And also, just for those that are listening, we'd really like to hear. This is kind of the first plant pathology themed webinar that we've done. So if you've got any suggestions for what you might like to see in the future, or criticisms, yeah, please jot them down into the comments box at the end here.
Okay, we're getting a couple of other questions coming in. So one question is whether you see cross-hybridization of different pathogen species.
Not typically.
Okay. I think that question is probably really relevant to when we might have used radioactive probe in order to look for similarities, but perhaps less common when we're using sequence data and blast.
Right. I mean, we certainly can get hybrids forming. Absolutely. It's possible, absolutely, but doesn't happen, all the time for sure. But there is certainly evidence of that happening in various systems, without a doubt.
So Martin, we have another question. So the question, first of all, starts by thanking you for the webinar, and then asks about your experience with isolating zoospores from the oomycetes.
Yeah. Okay. So that's something that we. Because of the size of this project that I was talking about, we really didn't want to do that approach. We didn't want to follow on that. We use that particular method. I know a lot of labs do that, especially for Phytophthora sojae. I'm familiar with some groups that do that. We just didn't want to get into that because of the size of the project, with having 3,000 isolates. And for that, I mean, really, it's just a matter of generating sporangia and getting zoospores released. And I have done this. That's right. So I have done that back in Australia. I did do a lot of work with, at the time, Phytophthora gnapodiides and Phytophthora megasperma, which has now been split up. But in that system, yeah, it can be a little bit finicky depending on the species that you're working with to generate sporangia and then actually get them to produce and release zoospores. There are a number of different tricks. Perhaps if that person wants to send me an email, I can find some information and send that on. Basically, he's using a lot of different nutrient media, liquid nutrient media to get production of sporangia, and then using like a sterile soil water extract to get sporangia to produce those zoospores and release them, so that we could do some of that sort of work where we could plate out individual zoospores.
Okay, great. So there's another question this time about the PCR primers. Yep. Are the same primers used for true fungi and oomycetes, or are there different fungi, different primer pairs?
Different primers. So what we typically do here in the lab, and what I've used a lot too, for the oomycete work that we've been doing, we've been using ITS6 and ITS4. There are a lot of primers out there, and you might need to potentially even tweak them depending on the genus that you're working with. With true fungi, I've typically used ITS1 paired with ITS4. There's plenty of information out there. You know, if you've got a particular group that you want to work with, just go and pull up some journal articles, and you'll soon be able to find some good primer pairs.
Okay, great. So the questions are trailing off now, and I notice that some of our attendees are leaving. So I think we're going to go ahead and wrap up. First of all, I'd like to thank everybody for your questions. As a reminder, a recording of this webinar will be available at eextension.org under the plant breeding and genomics pages, and that would be plant_breeding_genomics. Thank you so much for joining us again. Martin, thank you for your presentation, and thanks to the audience for coming. As a reminder, there will be a survey sent out. We really encourage you to fill out that survey. It helps us tailor the presentations that we put together. There is a place in that survey where you can indicate what topics you would like us to be covering. That's particularly important to today's webinar. It's our first foray into pathology, and so if you have specific comments that you would like Marty or others to cover, please put them into that survey. That will get compiled and get back to us, so we do rely on that information. So thank you again, and with that, we're going to close the webinar.